Hi, this is a Science Hangout from Science Mission. I'm Sadashiva Pai. Today, Dr. Jyoti Jaiswal with us. He's the Associate Professor in the Department of Genomics and Pediatrics at the George Washington University School of Medicine. Dr. Jaiswal has more than 40 publications to his credit here. He's a member of several societies and has mentored several individuals, reviewers for many journals also. And he has been awarded several grants to his credit. Let's welcome Dr. Jaiswal. Welcome, Dr. Jaiswal.
Thank you for inviting me, for giving me this opportunity. It was wonderful to speak with you and to the audience.
Thanks for joining Science Hangout. Let's get going to yourself, like your background, career, your education, everything.
So my training really is as a biologist, and I've trained in various forms of it, from plants to animals to microbes. I have really been interested in various sorts of technologies and their applications. So my initial training was in India. I did my master's from Baroda and then a PhD from the Institute of Sciences in Bangalore, focusing primarily on developmental biology, looking at organisms from the standpoint of cells to whole organism. And then got more interested in applying technologies, worked in the field of developing nanoparticles and other kinds of small molecule-based probes, fluorescent probes, for imaging purposes, and developing microscopy approaches for high-resolution imaging. And at that time, I sort of got interested in while I was at Rockefeller doing this work, Rockefeller University in New York, doing this work, I got interested in things that I've since pursued in my own career here at George Washington and initially at Rockefeller University.
Tell us about your lab members, like doing.
So the work that my lab is conducting right now really relies on a handful of people. It's about 6 to 8 people at any given time that my lab has. There are 2 graduate students and 2 postdocs, but that's a part of my group working on the projects that I'm primarily responsible for. Excuse me. Pardon me, I'm coming out of a cold, so you'll keep hearing me clearing my throat more frequently than I should normally. But so bear with me for those moments. The work that I do is, in addition to that, in highly collaborative in nature. So I collaborate with clinicians and basic scientists, as well as mouse geneticists and proteomics, omics people, with regards to developing the tools and techniques that we currently have to answer the questions about tissue injury and inflammation for disease purpose, for understanding diseases and for curing diseases. So there's a strong focus of my work at the moment on muscle diseases. But the work that I've been doing has been relevant or has been applied to brain injury, other kinds of tissue injuries as well. So we really are focusing towards developing different sorts of basic science understanding and preclinical and clinical applications of this towards understanding how chronic diseases related to tissue injury and repair deficits can be handled, understood, and treated.
And I know you are a busy person, writing grants, taking care of different projects and all. So can you talk to us about a little bit about your, other than the lab activities, like you do your hobbies, your interests?
So yeah, sure. So outside of the lab, really, I'm fascinated being a microscopist, which is one of the things that I do as a hobby, as a job, as a profession. I am very interested in capturing nature, so I enjoy photography, and I do quite a bit of that. I used to be primarily a landscape photographer before, but I've since then diversified also to taking pictures of the human natures, and I've really focused more on capturing moments rather than capturing videos. So I enjoy that quite a bit. I like hiking and go trip, both walking and jogging on trails, and so I enjoy going in nature quite a bit and capturing nature in that manner by using my camera.
Uh, okay. And can you talk us about any role models or any mentors you have in your career that helped you?
So I've
And
been very fortunate. Yeah, I've been very fortunate in having a series of very good mentors, and they've really been the reason why I am in the position that I am right now, being able to think and critically evaluate both mine and other people's work. So I owe a great deal to all of these mentors. I'm not going to start naming all of them, but from my undergrad days to my postdoctoral days, people who have mentored me and trained me have been very valuable in my own thinking and my ability to pursue science the way I do. But off late, I've realized I've found a new set of mentors as I have gone out of my educational phase to what people consider as the mentorship phase. I find that now the students are my mentors too. So I've been very fortunate to have wonderful students and postdocs, and the training I impart in them and the training they impart in me, I hope I can pursue to learn from and continue to aspire towards becoming better than what I am and what they would want for me to be in the days to come. So I think at the moment, I would say my biggest mentors are my students.
okay, that's nice to know. And let's talk about your work. And you have been doing some muscle-related work, muscular diseases, and all those things.
Yeah.
Can you please talk about some and work?
Sure. So I'll very briefly mention the interest that I developed in muscle diseases, and then we can go on to the scientific part of it. It really,
Okay.
I've always been a basic scientist, interested in treat by basic questions and developing techniques and technologies to address them. But during one of the visits, about over a decade ago now, to Children's Hospital, which is where I work now in Washington, D.C., I was taken by a conversation I had with patients and just the patients that were there as I was walking up to go give my talk on the 5th floor. And that sort of was one of the turning moments in my life where I realized that there is more to understanding science. There is more to it than just having a basic curiosity. There's more to it by way of application. And muscle diseases caught my interest for various reasons at that time. But it has ended up being a wonderful model for what it can teach us about tissue injury and repair, because it's very unique in its ability to both repair itself and regenerate completely, unlike most our tissues in our body. And it not only does that, but thrives on being injured and repairing from it. So it's a wonderful model system to study and understand the basic sciences of the tissue injury and repair process, cell injury, tissue injury, and repair process, and then apply it hopefully to other kinds of diseases, which we are finding, and I'll sort of touch upon that briefly in the talk itself, which I'm finding is the case, that discoveries that we are making with muscle model really is relevant to many other models, many other tissue models. So that's sort of my foray into muscle diseases, and I'm really hopeful that there is going to be a moment in my life when some of the work that we're doing can translate into clinic. And I'll give you a few examples of how we've really made inroads into being in clinic already with some of these things. I'm getting close to it.
Yeah, please go ahead and present your work.
Sure, that's it. So are you able to see my screen now?
No, not yet.
Okay. All right.
Yeah.
Is that better? Okay. So what I'll really talk about would be about cell survival and the mechanisms that are involved in the process of cell survival by using muscle as a model system. So what you see in the background of this is a image of a myofiber. But as I said, the things that you've discovered by using muscle tissue has been relevant to other cell models as well. So this is, you can sort of take most of it that I will describe today as relevant to many other cells. So let's just start with the very fundamental question of what happens to a cell in response to an injury of some sort. So this is sort of a cartoon showing how a mechanical trauma is disrupting the lipid bilayer. This is the plasma membrane bilayer. What that does to a cell in response to this trauma is you'll start to get influx of extracellular material inside the cell and efflux of cellular material outside the cell. So one of the major things that the plasma membrane provides a barrier for is iron and water. The influx of iron and water, ions like calcium, and efflux of materials like ATP, other kinds of cellular compartments, proteins, are both detrimental to the survival of the cell, but also to the functioning of the membrane itself. And this is sort of an example which illustrates some of the different events that can happen as a result of this, just from the act of calcium entry. Now, if you get excess water entry, the cell will explode. If you get excess calcium entry, you'll have all of these different pathways activated. So if not controlled, if this membrane damage is not controlled, that'll lead to the death of the cell. So much of what we talk about with regards to tissue injury really initiates at the level of a cell being injured. That's the first few minutes of tissue injury or cell injury. And what happens as a response to it then decides whether the tissue will degenerate or successfully repair from it. And the work that we've been doing really is addressing the question as to what happens to the cell and how the cell deals with this initial event, this impact leading to damage in plasma membrane, and copes with this damage by repairing it. So this is a question that people have thought about for a long time. This is not a novel idea. This is a quote from Victor Heilbrunn from 1930, where he says, "It is indeed a remarkable fact that protoplasm on emerging forms cells undergoes a reaction so essentially similar to that which blood undergoes when it emerges from a blood vessel." And that's where I've just captured that thought over here. He's drawing a parallel between blood clotting and protoplasm clotting, if you will. So that was one of the ideas that was thrown out, that protoplasm somehow gels as a result of damage to the plasma membrane, forming sort of a plug like the blood cells are forming here. But the current form in which we think of this process of repairing damage to the membrane is less focused on this clotting. Although no one's disproven it, it's not the mainstream thought. The mainstream thoughts are really from the work of Rick Steinhardt and Paul McNeil and a couple of other people who led this field back in the early 90s. It has identified a few different mechanisms or models, if you will, of how a damaged membrane can repair. If it's a small tear, the line tension on the lipids will allow the membrane to reseal back up quickly. If it's a larger tear, greater than a micron, then you will have these blue stripes, which are the actin filaments, and the black part, which is the membrane itself, coordinating by way of intracellular compartment vesicles to fuse and provide the additional membrane that will allow the damaged membrane to grow back and heal on itself, or form a patch that would then put a sort of a band-aid, if you will, on the hole and seal up the membrane. So there is evidence of batch formation from work in xenopasoocytes and other kinds of model. There's evidence from both mammalian cells and other kinds of cells that individual vesicles can fuse. But it's not clear if one or the other mechanism is what's being used. If these are the only mechanisms being used, is clotting another mechanism, or are there other events, other kinds of processes that go on that help a damaged cell membrane repair itself? So these are sort of the broad questions that need to be understood in order for us to know what kind of genetic or other cellular defects can lead to poor repair of damaged cells and then lead to tissue injury that becomes a chronic tissue damage. So that's been one of the focus of the work that we've been doing. Now, if you think of the tissues where the damage happens, there are a variety of mechanically active tissue where the cells are constantly damaged. You can think of lung, you can think of gut, you can think of cardiac tissue, skeletal muscle. All cells are capable and are prone to injury, but these are mechanically very active cells, so they get injured much more so than the others. So in this cartoon here, there's a depiction of how a cell membrane of myofiber or skeletal muscle fiber can get damaged as a result of activity, and what the models are with regards to what this myofiber will then do. So a few important things to keep in mind here, which I'll come back and refer to again later in the talk. So I'll just point them out also here. You have the extracellular matrix, which is the basement membrane, under which the blue line here, which is the plasma membrane of this myofiber, is in contact with through this protein complex called the dystrophin glycoprotein complex. It stabilizes by interaction of the extracellular matrix through plasma membrane to the myofibril, the element that contracts and allows the muscle fibers to contract upon stimulation. All of them to be in sync when there is a mechanical activity. If you lose the dystrophin protein, which leads to the disease called Duchenne Muscular Dystrophy, DMD, then you break the link between the outside and the inside, and then that can lead to increased tears in the membrane. I'll talk about this disease. There's another disease where you have this protein called dysferlin, which is thought to be involved in vesicle fusion or some sort of membrane remodeling event. If that's missing, then you can have these, remember the vesicle patches I told you about, they could be missing, and that could lead to poor repair. And there's an evidence for that from this paper about 15 years ago, from Kevin Campbell's lab, where he identified that myofibers that lack dysferlin repair poorly, leading to an increased amount of dye entry. The dye that is in this model, the laser is focused on these spots right here, and the injury in a healthy fiber prevents the dye from entering very rapidly, so you get a tiny little spot there. But if you injure either an absence of calcium, so the calcium is not there to trigger the repair response, then the healthy fiber fails to repair, and the dysferlin null fiber similarly fails to repair even when there is calcium. Even when the signal is coming in way of calcium, the fiber does not manage to repair very efficiently. So in diseases of muscle and other tissues, if the repair response is deficient, you will get more fiber death or more cell death. And that was sort of the crux of this paper, arguing as to how dysferlin might be involved in repairing the myofiber, and lack thereof leads to the disease, leads to this muscular dystrophy. So the same general architecture exists in other tissue types and for other cells too. And so you can sort of see the parallel between a repair pathway that might involve disruption of the membrane, basement membrane, plasma membrane, and internal compartment link, or lack of vesicles to fuse it to repair it. So work from one of the postdocs in the lab earlier before asked the question, if dysferlin is involved in the process of repair, how is it doing it? What is its role? Is what I described before with regards to its role as patch formation, is this the mechanism by which it's repairing, or there are other additional roles? Because the role of dysferlin per se has not been fully delineated yet as to what this is doing. These are all suggestions that it might be forming a patch. So what she did was she looked at cells from healthy donors and the limb-girdle 2B patients, the dysferlinopathy patients who lacked dysferlin protein, and looked at in the myoblasts, single cells. And the images here will show you roughly what's going on. But let me play the video to show you what the repair process looks like. Now, this is an assay I will repeat over and over again. It's the dye entry assay. The dye is FM143. I already referred to from Dr. Kevin Campbell's work before, that there is a laser injury. In this case, it'll happen at these spots marked by the green boxes. And as a result of injury, you'll get dye that's floating around in the media to start to enter the cell. And the entry will continue as long as the cell fails to repair, as the cell is repairing. Once the cell repairs, the dye entry stops, and that's why you'll see this plateau effect. A cell that fails to repair, and that's the example here where we've knocked down dysferlin protein in these cells here. The dye entry does not stop, and so you'll keep getting more dye into the cell. So this is a healthy cell. You get this injury, and you get tiny amount of dye entry here. There's a whole cell sitting over here, but the cell never really becomes green. Whereas if you do the same sort of an injury on a patient cell or dysferlin deficient patient cell, which is the example here, you will get, in response to injury, dye that keeps entering, and you can almost start to see the whole cell here because the dye is filling up inside and the cell is becoming more and more green. So this is the laser injury assay that tells us that these cells are incapable of repairing or poorly repairing in response to this injury. So the question that she went out after was, what is the mechanism behind it? What kind of vesicles or vesicular pathways might be involved in it? One of the things that earlier noticed was in dysferlin deficient cells, both the knockout cells from mouse or shRNA depleted cells or patient cells, there is a decrease in the number of lysosomes, which are one of the compartments that have been implicated in the process of repairing damaged cells. There is a decrease in number of lysosomes that appear at the cell surface, which is marked by the staining for the lysosomal marker LAMP1. So when we look at the dysferlin knockout cells, there's much less LAMP1 on the surface of the injured cells. So the injured cells are marked by red dye that has entered inside the cell, and the green dye is the immunostaining for LAMP1. So that's significantly reduced as a result of lack of dysferlin. So that sort of led us to the model that maybe vesicle fusion is reduced, and as a result of reduced vesicle fusion, we're not adding enough membrane, and that might be the reason for poor repair of the dysferlin patient cells. So what she then went on to do was to, and all of this work is published in this article, so that's why I'm glossing over many of the important technical details. But if in the question and answer session you have specific questions, we can talk about that. But I would recommend going over this article to get a more detailed technical idea behind these experiments. So what she did was she looked in live cells to ask, is this just a decrease in total number of lysosomes, or there's more to it? And one, and B, two, is there a deficit in patch formation? Are these vesicles truly forming a patch and plugging the hole, or is there something else going on? So what she did here was label lysosomes with a green dye, and by using a microscopy approach called total internal reflection fluorescence microscopy, which only illuminates about 200 nanometers of membrane, 100 to 200 nanometers of membrane that is in contact with the cell, with the cover slit, we can look at only at the membrane proximal events. So she's only looking here at the lysosomes that are sitting at the cell membrane. And by looking at individual vesicles, so this is the example here drawn for one vesicle, she could see if the vesicle fuses and releases content one at a time, or they all fuse together to first form a patch, and then the patch goes to the site of injury and plugs the hole. The expectation was with the patch, we will see one single fusion event, a bolus here, and if it's individual fusion events, we'll see them all spread all over the cell. So this video plays it out. You'll see the injury happen here in a few seconds, and then you'll see the response of the individual vesicles to it. So you'll see now the cell's been injured right there, and you start to see all of these vesicles, lysosomal vesicles, are fusing wherever they're sitting at. They don't coalesce to the site of injury and start to make a patch there. They just keep fusing wherever they're sitting at. And if she looks at the total number of lysosomes that fuse, individual lysosomes that fuse in response to injury, she found that was decreased by twofold. But surprisingly, the decrease was only in the first 15 seconds. The latter events were all about the same between the wild type and the knockout, dysferlin deficient and the dysferlin sufficient cells. The decrease was only in the first 15 seconds. So that was very puzzling. Why is the decrease only in the first 15 seconds relevant to repair? Is this causing poor repair? So one of the things that we observed with regards to this was as the amount of lysosomal fusion decreased in the dysferlin deficient cells, the amount of lysosomal enzyme released by the cell, called the acid sphingomyelinase, was also decreased. So there is a decreased fusion of lysosomes in the first 15 seconds that results in decreased secretion of acid sphingomyelinase, overall secretion. And that secretion deficit we thought might be relevant because it's acid sphingomyelinase is a lipid modifying enzyme. It acts on sphingomyelin. So we thought maybe there is something going on with regards to lipid modification that needs to happen within the first 15 seconds. And it's not a patch model. It's not a place where membranes will fuse and form a band-aid, but something they do something else by fusing rapidly in the first 15 seconds. Now, what we found in response to that line of thought was the sphingomyelinase secretion may really end up being relevant to the repair process. And if that is the case, then we might be able to reverse the deficit by providing the sphingomyelinase even before making the injury, so that the sphingomyelinase is available outside within the first 15 seconds when the lysosome fusion is deficient. So we took muscle fibers from mice that are deficient in dysferlin, so they're called the B6AJ mouse or Blage mouse, and we took the muscle fiber and injured them. If you look at the injury in a dysferlin deficient fiber, much like what Dr. Campbell's lab had shown, we saw dye entry that was significantly greater than the healthy muscle fibers. But then when we took the same muscle fibers and pre-treated them with sphingomyelinase, and this is stills from this video right here, which shows the amount of dye entry within the first 4 minutes of injury. If you took the same muscle and then pre-treated them with sphingomyelinase, you will see in response to injury, the amount of dye entry is significantly reduced, and the stills are over here. The plot here shows the amount of dye entry for a wild type in green, the dysferlin deficient fibers in pink, and the dysferlin deficient fibers that have been treated with sphingomyelinase in orange. So it turns out that the vesicle fusion is an important criteria or an important role for dysferlin in repairing vesicles, but it's not just the provision of membrane by the fusion of lysosomes to the membrane in response to injury, but it's actually the secretion of acid sphingomyelinase, deficiency in which causes poor repair. And if you can supplement it, this becomes a therapeutic approach. So this is one of the therapies that we've tested out in a preclinical model, and this is in the drug itself through collaboration with pharma is in for a different indication is in the clinical trial stage for that purpose. So if the preclinical models do end up proving this value for functional improvement, then the clinical trials, which is in phase 3 stage, will hopefully will allow us to then translate this quickly to the dysferlinopathic patients. So while we are doing this experiment, one of the things that we noticed in the laser injury assay for looking at the myofiber is the myofibers themselves, as they're repairing, and I'm going to play this video to show this point, as they're repairing, they secrete these tiny little vesicles from the site of injury. And hopefully you can catch them now, sort of running away from the site of injury. Here's one, here's another one, a whole bunch of them are running away now. So as the myofiber repairs, closes the wound, the membrane itself is being shed off, the damaged membrane is being shed off too. And this is another sort of neat way of looking at it, where we've taken the first image of this video and colored that green, and the last image of this video colored that red. By overlaying the pre-injury and post-repair images, you can kind of see all these red circles and vesicles that came out in the large red vesicles that have formed inside the damaged membrane. So the act of repair involves shedding of membrane. And there was a study that I will not talk about, which I've cited over here, where we've looked at role of these protein called the escort protein and endosomal sorting complex protein and recycling complex proteins that other people have identified to be involved in processes such as exosome formation in the multivesicular body, or HIV budding, or cytokinesis, places where a constricted membrane needs to be cut off. These proteins are used, escort proteins are used, and we found a role for those proteins in shedding of these vesicles, and inhibiting that ended up inhibiting repair of myofibers and myoblasts in culture. So not only do we need to secrete enzyme through lysosomal secretion to modify the membrane, but we also need to use these enzymes and other processes involving the escort complex to shed membrane that's damaged in order to allow the new membrane to heal and close the wound out. So this is a part that I've really glossed over very quickly. There's some interesting thoughts in this article, and I would refer your audience to go look at that if they're interested in the role of membrane shedding and exosomes and ectosomes in the act of membrane repair, but more importantly, also in the act of tissue inflammation and repair. Okay, so while we were doing this, and actually the escort work came out as a result of the proteomics stream that was initiated in my lab by Nimisha Sharma, who took this approach of purifying proteins from the cell membrane, or enriching proteins from cell membrane, and asking in a cell that is not injured versus a cell that is injured, what are other proteins that are showing up at the cell membrane? By doing that, we can then expand our palette of intracellular compartments beyond just lysosomes, which were the only compartment that people had talked about in the literature to be involved in repair. So we wanted to actually find out if that is it, or if there are other compartments that are involved in the repair process, and if there are other processes involved in repairing damaged cells. So in this strategy, what she did was she pulled, by using a biotin tag for the cell membrane proteins, she pulled down membrane proteins from these two populations of cells and ran them on a mass spectrometry to compare what changed from an uninjured to an injured cell. She identified in the myoblasts the escort proteins, but in the myofiber, her work showed that the bulk of the proteins that we are pulling down are mitochondrial proteins. And that was very puzzling at that time. We thought, why would mitochondria show up at the damaged cell membrane? But digging back at the literature, it turned out this was not really a surprise. This is just something that was not very heavily investigated, but has been reported back as early as 1990s, where work from Miranda Grant's lab in Australia showed that when she would look at EM images of damaged myofibers, she would see these stumps, or the places where the myofiber heals itself, is completely full of mitochondria that is not damaged. They even have neatly organized cristae. And these mitochondria, seemingly functional mitochondria at the stump, was a consistent feature. This is in rat muscle, but in other muscles too. So we went back and asked, is this something that's happening? These were images taken several hours after injury. So it wasn't clear if these mitochondria accumulated at the site of injury later in life of the myofiber after the repair or during the course of repair. So we asked ourselves this question: is mitochondria, as our mass spec data is showing us, is mitochondria accumulating at the site of injury right after the injury itself? So we took the advantage of live imaging of mitochondria and myofibers to do so, and this is showing you just a couple of examples from stills as to how a myofiber looks before injury, and that is stained for mitochondrial marker, and you can see mitochondrial accumulation right at the site of injury upon injury. And this video illustrates that using the same approach, the myofiber is labeled with mitochondrial dye to label all mitochondria that are active, and then there'll be a laser injury right here to show what the fate of the mitochondria is. You'll see in response to repeated injury, you get this accumulation of mitochondria at that site. And while there is, as the fiber moves, the mitochondrial fluorescence all over seems to be increasing, the increase really is greatest at this site. So Nimisha reported that in an article a few years ago, and what she also showed in the same article was if you were to depolarize mitochondria by using this mitochondrial uncoupler, protonophore CCCP, you'll get a dose-dependent inhibition of repair, as indicated by the same FM dye fluorescence increase assay that I described earlier. So at 5 micromolar versus 10 micromolar. And at 10 micromolar, the repair is almost as bad as the repair in absence of calcium. So calcium is an important signal for repair process, and this is a demonstration that depolarizing mitochondria can give the same level or similar level of deficit as minus calcium scenario does. So in response to injury, you see, as before, nice containment of dye entry in a healthy fiber, but when you treat them with CCCP, the dye fully fills up the myofiber. So it sort of remained as a puzzle for some time as to what the mechanism behind this mitochondrial involvement of repair is. Adam Horn, a graduate student in the lab, took up this project and decided to investigate what the role of mitochondria might be in the repair process. So the first thing that he saw was, in response to injury, the mitochondria that are at the site of injury rapidly take up calcium. So this is a calcium sensor that is genetically encoded calcium sensor called mitochondrial gecko. In response to injury, the calcium increases here and all throughout the cell, gradually all throughout the cell as well. But later on, the calcium drops in the back of the cell, whereas it continues to stay high at the site of injury. So this is sort of a trace of that at the site of injury. You get this increase and a gradual decrease, while the mitochondria in the back will fully drop down by this time to the basal calcium before injury. The mitochondria here don't do that. So at the site of injury, since mitochondria is increasing in calcium, he asked, could mitochondria be using this calcium increase to initiate some repair process? One of the key role of mitochondrial calcium, increasing mitochondrial calcium, as this diagram shows here, is to activate the ATP synthesis, the synthetic pathway, by acting at different steps along the cycle. So we thought maybe mitochondria at the site of injury are taking up calcium and are triggering calcium-induced ATP production. And since it's an energy-intensive process, maybe ATP is being produced to facilitate repair. And Adam looked at that, and I'll get to the ATP data in a second. Before that, I guess I should just touch upon the importance of calcium itself. And to look at the role of calcium itself in the process of repair, what he did was he looked at cells that are deficient or that lack calcium transporter, MCU, mitochondrial calcium unipolar knockout, and saw that clearly, as expected, these cells don't take up calcium in response to injury. So there's a significant reduction in their ability to take up calcium in response to injury. And what that does, it causes the cytosolic calcium to increase and stay up longer. So this is a calcium sensor for the cytosolic fluid 4. If you take cells that are MCU knockout and look at its calcium in the cytosol, it stays up higher for a longer period of time and to a greater extent. And if you then look at the repairability of these cells by using the same FM dye assay, whereas the wild type repair in the first 50 seconds, the MCU knockout continue dye entry and there is about 3-fold increase in the number of MCU knockout cells that fail to repair. And that indicated to us that this calcium uptake ability of the cell is critically linked to the repair process. So back to the idea of ATP, if ATP is required for this process, Adam took the approach of inhibiting ATP production first by using either an inhibitor of glycolysis, 2-oxy-D-glucose, or by using azide to inhibit mitochondrial respiration, or by using a combination of those two drugs to inhibit acute ATP production. So these cells have just been acutely treated with these drugs to block ATP production at the time of repair, not chronically, just at the time of repair. And turns out it doesn't matter whether we treat them with one or other or a combination of them, there is no difference in the repairability of these cells. So that was a little surprising because we thought acute ATP production must be needed for the process of repair, but turns out it is the basal ATP that exists inside of a cell is sufficient to repair the cell. Even if the cell does not have acute increase in ATP production, it repairs just fine. So then what could the role of mitochondria be? So if I can go back to this cartoon for a second, as a result of this increase in the cycle, electron transport chain, while you get ATP as the main product, one of the byproducts of this process as a result of the electron carriers donating their electron and causing oxidation is production of reactive oxygen species. So we then asked ourselves, is there an increase in reactive oxygen species production in response to injury by these mitochondria at the site of repair? So Adam took redox sensor and monitored the level of mitochondrial ROS, a redox enzyme that is targeted to mitochondria, and looked at, called mitoxox, and looked at the redox level at the site of injury and away from the site of injury. And sure enough, in response to injury, you get this heat map which shows a burst of mitochondrial ROS that continues to increase as the cell tries to repair itself. So keep this in mind. This is fairly consistent with what we saw at the site of injury. The mitochondrial calcium goes up and stays up, and you're getting this mitochondrial ROS going up and staying up as well. So we asked, is this really important, or is this relevant for repair even? So Adam took this drug rotenone that blocks complex 1 and causes excessive matrix mitochondrial ROS production, and saw with this or another drug called antimycin, which does the same job at a different complex 3, but they both ended up causing greater mitoxox production. And shown here is the example of rotenone actually caused the FM dye entry to stop earlier than the healthy cells. So in the untreated, you get greater dye entry than in a rotenone-treated, acutely treated cell. So transiently increasing mitochondrial ROS production was actually beneficial. Unlike the ATP production, ROS production was consequential for repair process. And he did the reverse experiment where he put a drug called mitotempo to quench the mitochondrial ROS, so that whatever mitochondrial MCU-mediated mitochondrial ROS through calcium would get produced will get quenched rapidly by this drug. And that reversed the phenotype, caused increased dye entry, and the mitoxox production was significantly down in this scenario. So just by increasing ROS or by decreasing ROS in the mitochondria, it appears that it's the mitochondrial ROS that the calcium is increasing, which is facilitating repair, not the ATP itself. So this is interesting because it was, again, very counterintuitive. ROS is thought to be in muscle and other tissues, thought to be a detrimental signal. It's something that causes damage. But in this acute model, it actually appears to be beneficial for the process of cell survival. So we asked, what might ROS be doing? And one of the things that I mentioned in the model before from McNeil, Steinhardt, and others' work, and work of Bill DeMent's lab, is that you get rapid actin depolymerization and polymerization at the site of injury. And their work suggests that this might facilitate patch formation, but the act of actin depolymerization and repolymerization has not been fully investigated as to what the mechanisms there might be behind it. So one of the things that mitochondrial ROS has been shown to do in literature is to alter actin polymerization. So we first asked, can we see actin polymerization and depolymerization, and is that dependent on mitochondrial calcium uptake? So this is using F-actin reporter called LifeAct. And Cherry, if you just look at this zoom in of this region of the cell as it undergoes repair, you get a large burst of F-actin accumulation loss, and then burst of F-actin accumulation at the site of repair. And if you take the MCU knockout cells, they are clearly compromised. They show the drop in F-actin as a result of injury, but they never build it up as the wild type does. Not only do we see this correlation with the calcium uptake, but by using the same agents, antimycin and mitotempo, to increase mitochondrial ROS or decrease mitochondrial ROS in case of mitotempo, we can mimic the phenotype. So he was able to show that antimycin caused a slightly greater increase in amount of actin accumulating at the site of injury, whereas mitotempo quenched it. So there is a need for calcium to go in, and upon the calcium entry into the mitochondria, there is then a subsequent increase for mROS, mitochondrial ROS, to be formed in order for the repair to happen in a healthy cell. And we can enhance that further by acutely increasing the amount of ROS, or we can prevent it by completely depleting mitochondrial ROS by using drugs. So what might the mechanism be? Well, how could mROS be helping with repairs? There's evidence from Keith Berridge's lab showing that ROS activates ROA by acting on a specific cysteine residue, oxidizing the cysteine residue, which locks this molecule in an active state without a GTPase cycle. So we thought maybe ROA is getting activated at the site of injury, so we use this ROA flare sensor which is a threat-based sensor at reporting on the ROA activity. In a resting cell, you get minimal activity in the lamellipodia, the inactive lamellipodia. But once you injure the cell at any of these surfaces, you see a rapid burst within 10 to 30 seconds, a burst of ROA activity, which is quantified here. And this ROA activity is quenched, is blocked if you quench the mitochondrial ROS by using MitoTempo. So it appears that the sequence of events is calcium entry leading to mROS production leading to ROA activation, and ROA is a direct regulator of actin polymerization. So I just quickly wanted to check this, if this pathway, which we primarily worked out in a mononucleate cell, also holds true in myofiber, which are multinucleated cells with a very different architecture. And for that, what he did was he looked in this mouse that expresses life act in the muscle fibers and labeled for the mitochondrial ROS by using mitosocs, and simultaneously imaged in the same fiber what is the status of mitochondrial ROS production and actin accumulation. And as you can see from this merge image, you get actin accumulation right around the region where there is an accumulation of mitochondrial ROS production. And they, if you look at just over time, they pattern almost identically to each other. And again, the same suspects, antimycin and MitoTempo, activator of mitochondrial ROS and quencher of mitochondrial ROS, are able to show the opposing effects on actin accumulation and the ability of the fibers to repair. So when you quench it with MitoTempo, there's about 2 to 3-fold increase in number of fibers that fail to repair. So the results sort of hold true from single cells to multinucleated myofibers. They seem to be using mitochondria for the process of signaling locally for the purpose of actin accumulation and facilitating repair. So another grad student, Maria Villa, in working in collaboration with Dr. Nagaraju, a longtime collaborator at Children's, got interested in this observation of Adam because what she was looking at was this disease I told you about earlier, Duchenne muscular dystrophy, by using a mouse model called MDX mouse, where lack of dystrophin causes myofiber damage and poor repair. So what her data was showing was these MDX mice had reduced mitochondrial activity. This is cytochrome C activity, and also reduced mitochondrial content as measured by mitochondrial structural proteins TOM20 and VDAC, which shows all reduction in it. So we started looking at how the deficit in mitochondria might be manifesting in these Duchenne mouse model fibers. So what we looked at was the response of mitochondria in the MDX muscle. And you can see in response to injury in the healthy muscle, you get this robust increase, and this mitochondria in the disease muscle, the Duchenne mouse model muscles, they don't accumulate as much. There's a significant reduction in how much mitochondria manages to respond to injury. And on top of that, what she was able to see was the other proteins, dysferlin and the interacting proteins, annexins and caveolin, they tend to increase as the disease progresses or disease initiates in the MDX model. These are two different DMD model, MDX with one allele and another allele, both lacking dystrophin expression. In both cases, as you see, the dystrophin is missing, the amount of dysferlin goes up, amount of annexin 1, annexin 2, caveolin, they all seem to be going up. So the vesicle-mediated repair pathway is upregulated in this mouse fibers, whereas the mitochondria is downregulated. So this gave us a nice model to ask the question, is vesicle-mediated repair pathway the primary repair pathway for repairing myofibers, or does mitochondria have a role to play? So since these are upregulated, if this is the most important mechanism for repairing myofibers, we should get an improved repair of these myofibers. If mitochondria has a more important role to play, then we should see a reduced repair of myofibers. So she went ahead and checked that. She looked for what the status of the myofiber repair. And this is, again, videos and stills from there showing that healthy myofiber, the diseased myofiber, the MDX52, takes up a lot more dye. And I'm just going to play this video because it's just so dramatic with regards to how much dye it takes up. And as a result of the dystrophin deficiency itself, the fiber hypercontracts more violently than even the dysferlin one that I showed you before. And she went on to check this further, not only by laser injury, but also by using eccentric exercise. So she took muscles out and exercised them and looked at loss in muscle force in response to 1, 2, 3, 4, and so on, multiple muscle contractions. And so the MDX seems to drop much faster compared to the wild type and takes up a lot more dye, another membrane impermeable dye called protein orange. It takes up a lot more of that, even in response to one or multiple contractions. So the myofiber clearly seems to be getting its membranes, plasma membrane, damaged as a result of either contraction or laser injury, and getting greater damage and allowing greater dye entry than the healthy muscle fibers. So, and you can see that the number of fibers that manage to repair significantly reduced when there's dystrophin deficiency. So it sort of is leading us to now believe that in case of dystrophin deficiency, it's not upregulation of vesicle repair that will improve the disease, but upregulation of the mitochondrial pathology, or downregulation of pathology and upregulation of mitochondrial function that might facilitate repair. So we haven't gone on to test any therapies yet. These are very recent findings, but we are now in the process of trying to look at how we can help just like we did for the dysferlin fibers, how can we actually help the Duchenne fibers repair better? So just quickly to summarize then, really what I've been telling you is I started off by showing you a model regarding membrane repair that was limited to vesicle-mediated either patching or provision of extra membrane. We've added since then, and others have contributed significantly to it, other modes of repair, either by way of actin accumulation. Annexin, I didn't have the time to tell you the story, but we've looked at the role of various annexins in the process of repair, and annexins have been implicated in the process of repair. This is from an article by Noah Weisleiter's group, that a nice review summarizing various models of it, which came out in 2015. And it also talks about the escort and the acid sphingomyelinase as two important regulators, either of endocytosis or ectocytosis, facilitating membrane remodeling for the repair process, and synaptotagmin or snare-mediated repair pathway, which we've looked at in astrocytes, but we've not dwelled much into it in this talk. I didn't talk about that either. But all of these are sort of complementary pathways that facilitate the stabilization, if you will, of the membrane, of the damaged membrane for the process of repair. In addition to that, what I've spent some time telling you about is the role of mitochondria that dovetails into this actin and other processes by way of signaling. So you have a signaling process that goes on in the repair, and then you have all these structural processes that need to go on for the repair to happen. And so this was not covered there because we published this more recently, but what I would like to sort of leave you with regards to the models is this may not be the end of it. We are still very actively in the process of discovering, we and others are actively in the process of discovering what other pathways might be involved in the repair process. And just from realizing there are these multiple different pathways, I've given you an example of one disease, Duchenne muscular dystrophy, and another one, dysferlin deficiency or limb-girdle muscular dystrophy 2B, where vesicle-mediated or mitochondria-mediated repair pathways might be deficient, and hence can lead us to therapy. And so the last sort of closing slide that I'll give you with regards to the data itself, summary of the data, is this model where you have, in case of healthy muscle, you have vesicle fusion, not necessarily to form a patch, but to release this enzyme, acid sphingomyelinase, lack of which, when the dysferlin, these blue cylinders are missing, lack of which causes reduced secretion, and we can rescue it by providing sphingomyelinase from the outside, which then will facilitate these fibers to repair efficiently. Or in case of dystrophin deficiency, where the mitochondria themselves are dysfunctional because of lack of dystrophin and excess calcium influx. And that might set in this poor repair, leading to more calcium influx, leading to greater pathology and so on, which causes this positive feedback, causes greater myofiber death, even when the annexin and dysferlin-mediated vesicle repair pathway is upregulated in these fibers. And so therapies that target these mitochondria, dysfunctional mitochondria, might be beneficial. And there is evidence in literature that that is the case. Improving mitochondrial function does, or production by way of PGC1 alpha is beneficial in case of the MTX mouse model. So I'll end by, I don't need to go over the summary again since I've already done this pictorially, but by thanking people who've really been instrumental for all this work. While I'm presenting it to it, it's the work of all of these people. I've tried mentioning their name as I went along for people whose work I was able to present today. But for others whose work I was not able to present, have all contributed significantly along with the collaborators, many of them very long-standing collaborators, that has resulted in the both basic science understanding, but also the therapeutic development. And all of this work is supported really by NIH and private foundations, including the Muscular Dystrophy Association, that just supported the work, our work on looking at the therapy for dysferrinopathy, and the grant from NEAPS that's now supporting this work even further. So thank you.
Thanks, Dr. Jaiswal. So if you can unshare the presentation. It's a really interesting presentation. You have summed up pretty well with the different the repair of the question regarding the mitochondrial movement. Is there any signal? What makes mitochondria move to the injury site? Is there any signal which injury, or is it the calcium which is drawing the mitochondria to the site? And the question would be, is it the whatever the mitochondria within the cell, or is there any of mitochondria happening?
So let me start with the housekeeping question. Can you see my screen already? Can you see me?
Yeah.
Okay, good. So let me answer both of your questions.
Yep.
Let's start with the first one. What causes mitochondria to accumulate at the site of injury? So the way the myofiber is organized, the myofiber contracts, the myofibrils contract as a result of calcium increase. And the calcium in this situation, the injury is coming from the tear in the membrane, the damage in the membrane, which causes the myofibrils at the site of damage to contract. The mitochondria and muscle fiber are laid along the myofibril. So as the myofibril contract, all the mitochondria get pulled together with it. And that provides a very rapid mechanism for myofibers to accumulate the mitochondria at the injury site. It does not require a motor-driven transport, which will be far too slow for this process. So that's the mechanism for mitochondrial accumulation. Now, once it's there, I've already outlined the various steps through which it undergoes the repair process.
Okay. And regarding the lysosomes and all those things, how do they, in terms of mitochondria, lysosomes, exosomes, and all those things?
So
separate.
Right. So let's take them a step at a time. So if you think of lysosomes, what it appears to be doing is not only providing membrane, but more importantly, providing the enzyme sphingomyelinase to the outside of the cell to remodel the membrane, to change the membrane stability, fluidity, and allow the lipid mobility to facilitate the closure of the wound. The ectosomes or the exosomes are primarily involved in shedding membrane that is damaged. So as soon as you tear the membrane up through some sort of a mechanical or chemical trauma, most likely all of those oxidative events through mitochondria and others that are happening will oxidize the lipid. Oxidized lipids cannot then be used to close a wound because that will destabilize membrane. So you need to get rid of that. And ectosomes and exosomes that are forming there will facilitate the process of getting rid of that lipid and then allowing the remaining healthy lipids, if you will, to close up the wound as the process of actin-mediated repair allows the membrane to be constricted back in. So they seem to be doing
technology.
Roles.
Go ahead.
Go ahead, Stasha.
With the current imaging technology, simultaneously look at lysosomes, mitochondria, by labeling these things at a time, same time?
So we've looked at, we can do multiple different probes simultaneously. So I showed you a few examples where we looked at actin and ROS. We've looked at mitochondria and actin. So we've done lysosomes with other compartments, other endosomes and other things. So yes, so we can look at multiple different compartments simultaneously. The nice thing is that the repair response is so robust and so precise with regards to its timing that oftentimes we don't even have to do that. We don't even have to look at every single player all at the same time. We can do them sequentially. And since the repair happens in this very strict 10 to 60 second time window, typically, we can time them. And so in the S-Corp paper that I did not have the time to go over, we've actually timed when does the first molecule, second molecule, and the third molecule come in the process of recruitment of this entire S-Corp complex. And just by looking at the timing process, we were able to say who's the initiator and who's the follower. And then by doing siRNA-mediated knockouts, we were able to confirm that independently. So the whole process is fairly stereotyped and very strict in its temporal manner, allowing us to do this either in combination or independently and then combining it together.
Thank you so much. I think we are just over the time right now, and I really appreciate you coming in spite of your throat and so I really appreciate that. And if you have some good results, shoot an email and we'll get back and have another hangout sometime. So again, thank you so much. I appreciate your time.
Thank you so much for the interest and for this opportunity. Have a wonderful day.